Thank you for joining us today for our second webinar that focuses on improving immunohistochemistry and immunofluorescence workflows. Today's webinar is about positive and negative controls in IHC and IF applications. This will be presented by my colleague, Dr. Craig Powell. Following the 20-minute presentation, we will have 10 minutes for a Q&A session. Please submit any questions you may have using the chat function on the GoToWebinar site, and we will answer your questions. I am now pleased to introduce you my colleague, Dr. Craig Powell. Over to you, Craig.
Thanks, Byron. Hello, everyone. Good morning, good afternoon, and good evening, wherever you may be located around the globe. Thank you for joining us for this second webinar in our series. As Byron mentioned, today's topic is all about controls. So a brief overview, we're going to be looking at the types of controls you'll encounter with your immunohistochemistry and immunofluorescent applications. Then we'll take a deeper dive into positive controls and negative controls, particularly what are they, why use them, and what are some examples of appropriate controls that you could implement into your workflows. So 3 types of controls that you'll encounter in your assays. The first one I've got here is a defined deletion control. It's a type of negative control that omits defined parts of the workflow to help identify the source or sources of background that you may encounter if you do see non-specific staining. So this is a troubleshooting methodology. Now, this was the focus of our first webinar about 3 weeks ago. So if that's of interest to you, certainly I'd recommend going back and revisiting that webinar on our website after this webinar, of course. So for this webinar going forward, we'll be looking at positive controls that help define assay sensitivity and specificity, and also look at negative controls that help identify false positive staining that may occur in your preparation. Let me just deviate here a little bit and tell you why we put this webinar together and why we feel that controls are an important topic. We do encounter investigators who do apply these controls, positive and negative, infrequently and or inappropriately. And you know what this leads to, unfortunately, is non-reproducibility by them, or certainly by others who want to build on their research. And that's certainly disappointing. You know, without the appropriate positive and negative controls in place, the staining that's achieved on a test specimen is essentially meaningless. So therefore, the idea of this webinar is to showcase that these controls are to be used together, they're complementary, they're synergistic, and they allow you then to make important observations and extrapolate nice, clean data from your test specimen. Okay, so let's get into positive control sections. What is it I'm talking about when I refer to a positive control section? You know, a section that's known to contain or express a moderate to high level of target antigen. And I've got an example here of a B cell marker, CD20, in tonsil. It's showcased by the image here of a nice dark DAB staining where you'd expect it to be. Certainly, this would be an appropriate control or positive control for this antigen in this tissue. I've got another example there of the Chi67 or Ki67. You could also use that in tonsil. And I've indicated both of these here to show that for investigators who may be concerned about costs or trying to source multiple positive control blocks of tissue or sections, indeed, you could use the same block of tissue for multiple target antigens that you're looking for in a lot of cases. So that may help offset some concerns investigators have about positive control sections. Now, as showcased here in that image, positive control sections incorporate the entire detection methodology, which is obviously from antigen retrieval, your quenching, your blocking, primary antibody, and then the rest of the detection reagents, indeed, whether it be enzymatic or fluorescence-based. Now, I do have a little asterisk up by the definition I've given there, and we'll revisit that a little later on in the presentation. There is an exception to that definition that I'll explain a little later, so just keep that in mind. So why use positive controls? Certainly to validate the assay you're running, whether it be immunohistochemistry or immunofluorescence. Importantly, what you're looking for is specificity of cell type, and indeed specificity of the cell compartment. If you are looking for a nuclear expressed antigen, you want to see staining in the nucleus, not in the cytosolic area or the membrane. Another reason for running a positive control is to identify any off-target, non-specific binding that may occur. And certainly this gives you then an opportunity to then optimize conditions to get a good signal to noise ratio so you can move forward without ambiguity in your test specimen. And if you are introducing, say, a new or a novel primary antibody or different detection reagents, let's say you're swapping from a peroxidase to an alkaline phosphatase system, something like that, you'd also want to use a positive control where you can work up these reagents to get a good signal. The final point I've got here is periodic assessment to ensure reliability of the test results in accordance with the assay parameters. And essentially what I mean by that is if you do get unusual staining in the test specimen or atypical staining, you may start to question, gee whiz, is this a great finding? Is this something I should report on? And this is where a positive control should be run in parallel, and that way it gives you confidence on whether or not that particular observation is in fact accurate or true, or maybe artifact for some reason. Another key reason for running a positive control is in the circumstance where in fact you may obtain no staining or complete absence of staining. As I've shown here, you might be ending up scratching your head saying, gee whiz, what's gone wrong? And indeed, I've fielded inquiries like this from investigators saying, you know, I've had some great staining the last couple of months, everything's been going great. And this last week, I've got nothing, not even background staining, nothing, no trace staining. And they're saying, gee whiz, did I leave off the enzyme substrate? Is it not working? Did I use the right primary antibody? Did I use the right dilution? Did I not do unmasking? And you know, they're just arbitrarily swapping things out and going down a wrong path, spending time, wasting money trying to find out where they went wrong. This is where a positive control is invaluable to help avoid all of that drama and cost. And you'd certainly then use the positive control. Did that work? Did that give appropriate staining? Therefore, you can identify which parts went amiss and may need to be refactored in your test specimen. Okay, some considerations when using a positive control. Sections do not have to be from the same organ or tissue type, or indeed the same species as the test specimen. I did give the example earlier of the Ki67 or the Chi67 in tonsil. That is certainly expressed in a wide variety of tissues, that mitotic marker in skin, in gut, in a lot of tissues. So indeed that example hopefully showcases that you can use different tissue types as positive controls. If you were going to use a different species as the test specimen, just make sure the primary antibody does recognize the antigen in that alternative species. Now, sections do have to be treated in the same manner as the test specimen. So the example I've got here in the image is a nice specimen in a paraffin embedded block. So indeed, if your test specimen is, say, formalin fixed paraffin embedded, you'd want to make sure that the positive control material is also formalin fixed and paraffin embedded. Conversely, if the test specimen is acetone fixed, frozen, embedded in OCT, you'd want to make sure that the positive control is also acetone fixed, frozen, embedded in OCT. The idea is to limit the number of variables here so you can showcase or run these in parallel and be confident about the results you get. The last bullet point I've got here on this slide is whether you may be conducting multiplexing, multiple antigen labeling. What do you do in those scenarios? And what I'd recommend would be to set up known positive controls for each of those target antigens you're looking for, whether it be 2 or 3 or however many you're trying to identify in the one tissue section. This will alleviate potential issues you may have down the road. Okay, so missteps in applying a positive control. Well, certainly, as I've indicated here, not using one. And, I think that the example I showed before of the absence of staining on the test specimen speaks volumes and should resonate with some investigators who have actually experienced that firsthand. You can spend days, weeks, months trying to troubleshoot this when in fact a strong positive control in place would have alleviated all of those headaches. The second bullet point I've got here is a lot of investigators really love to go out and try and find their results really quickly. Hey, let's see what staining we've got in this novel specimen, whatever it might be, and they use the test specimen as a substitute for the positive control. The argument I've got there is, while that's well and good, they may not be seeing the full extent of expression. If they'd worked it up on a positive control, maybe they'd see some really neat staining in a population of cells or whatever it may be that's not seen in their test specimen. And unfortunately, they're not getting the full bang for their buck, so to speak, in that test specimen, particularly if they aren't seeing the novel staining that they would have seen if they'd run an appropriate control. Now, if you remember the little asterisk I had in the definition in one of the first slides I showed about, a known section or to express moderate to high levels of target antigen, the exception here, this is where we're revisiting this asterisk, would be if the antigen is only present or assumed to be present in that test specimen. And in that case, you're thinking, gee, what do I do here? I think it's there, but I'm not sure. So this is where you'd want to look at the test specimen and see if there may be some other tissue types that you can target for a positive control. For example, let's just say in this test specimen, you've got endothelial cells that you're looking at nitric oxide or whatever it might be there. That's what you're trying to target, but you don't know, because it's a treated specimen, what the expression will be. Are there any epithelial cells where you could use a cytokeratin? Are there any smooth muscle cells in that same section where you could use an actin or a desmin or something like that, that you could then use a primary antibody of the same species as that target antigen, as the same isotype, and use the same secondary detection reagents to then show that all of those are working in that test specimen, that it's been antigen retrieved appropriately, it hasn't been overfixed, those sort of variables. So what you want to do is limit those variables and then apply that primary antibody to that test specimen. Hopefully this gives you an insight into how that may work. Okay, let's move on now. We'll pivot over to negative control sections. So what is a negative control? Certainly a section that's intended to show an absence or no staining whatsoever. And I've got an example here of a tissue section just showing some hematoxylin nuclear staining. So the idea of using a negative control is to ensure that the detection reagents, which include the primary antibody, are not generating false positive data. Now, a lot of investigators may say, you know what, Craig, we're set on negative controls. We just simply omit the primary antibody. People have published on this. And I've got a couple of quotes here from journals that certainly do highlight that. I'm not picking on JBC, I'm not picking on osteoarthritis and cartilage journals, I'm not picking on these papers or the authors who wrote this, but this negative control that they've openly said all they're doing is omitting the primary or secondary antibodies aren't adequate in the sense of addressing whether or not the primary antibody may be inadvertently binding to some tissue components, FC receptors, whatever it might be, or some other, residues that may be in there and generating, false positive data. This is an opportunity, and this is why, one of the reasons at least why, I put this together, this webinar. I think there's an opportunity to do some better science here. And simply omitting the primary antibody is not what I'd refer to as a publication-worthy negative control. Anyone who's presenting this in a scientific endeavor, at writing an abstract for a paper, whatever it might be, you want to be very confident that what you're describing in your observations is in fact accurate. Okay, so this raises the question, okay, what is an appropriate negative control? And the one I've got here, substituting a primary antibody for a non-immune immunoglobulin, is probably the most widely used and easiest to implement. The example I've got here is if your primary antibody is a mouse IgG primary antibody. It might be mouse anti, as I've used before here, Ki67, for example. So the idea would be to substitute in a non-immune mouse IgG at the same concentration and the same parameters as that primary antibody. Then you put on the rest of the detection reagents. The idea is then to see that an absence or no staining on that tissue preparation. Now, in some instances, I will say this non-immune mouse IgG, is probably a pool of mouse IgG 1, 2A, 2B, etc. Sometimes you may get some tone-in or background at the same concentration as the primary antibody. If you do encounter that, you may want to have a closer look at that primary antibody. Indeed, if it's something like a mouse IgG2A isotype, you could then use a more defined substitute like a non-immune mouse IgG2A. That'll eliminate all of the IgG1, 2B, etc. and give you more of a defined negative control and should hopefully clean up that trace background or toning that you might see with the example I used there in point A. Again, though, you would, with this substitute of the non-immune mouse IgG2A, you'd use it the same concentrations as the primary antibody and under the same conditions as in temperature, timer on the tissue, etc. Another appropriate negative control is pre-absorption of the primary antibody with the immunogen used to generate the primary antibody. And I've got a little cartoon here demonstrating the primary antibody binding the immunogen in solution, let it incubate for a given period of time. Indeed, some investigators may even say overnight at 4 degrees would be appropriate. And then putting that complex on the tissue preparation along with the secondary detection reagents to see an absence of staining. Two things with this negative control. Number one, more than likely if you're getting that primary antibody from a commercial vendor or commercial source, they're not going to make that immunogen openly available. I understand that, and therefore this particular negative control may not be available to a lot of investigators, the vast majority of investigators out there. That's fair enough. The other point with this particular negative control is that some investigators have expressed concern that the primary antibody, that while it does bind the immunogen in solution, isn't a true representation of how the primary antibody may bind or what it may recognize on maybe a heterogeneous piece of tissue, a tumor sample, whatever it might be that you're looking for. And, to that, I certainly acknowledge that point. But this in itself is not supposed to be validation of the primary antibody. This is not an experiment in a vacuum. This is supposed to be done in combination with other data, whether it be a Western blot or other validation data, ELISAs, column affinity purification, that has something behind it that you can use in combination with this negative control. Use those together to show an absence of staining. So if it's possible to get the immunogen, maybe under a condition of a synthetic peptide, maybe that might be better. or a more valid option, you can certainly use this as a negative control. A further appropriate negative control would be to use an irrelevant primary antibody, something that doesn't bind to the tissue that you're working on. And I've got that little image there of off-target binding, as in not the target you're looking for, and indeed nothing in the tissue. So what is it I'm referring to as an irrelevant antibody? Let's just say you're working on CNS tissue or brain or spinal cord tissue. You're pretty sure that, stratified keratinized epithelium is not going to be present in that tissue. Therefore, use an antibody against an antigen, a cytokeratin, that might be only present in stratified keratinized epithelium on that brain tissue. The idea being to use it matching the same species that you intend to use for your target antigen and the same isotype. And the idea here is to show an absence of binding of that species isotype on the tissue with the same detection reagents. So it's irrelevant. It's not going to bind to that tissue type. And I think that this is also an appropriate control that you could implement. One further negative control I've got here is removal or absence of the target antigen. The example I've got is digestion. In some cases, use of an enzyme, like if you're looking for collagen, you could use a collagenase, gobble up all of that collagen. So when you put on your anti-collagen antibody and detection reagents, it's not there, you won't see staining. I understand that may not be an option for a lot of investigators. You can't really digest out a cytokine or something like that. In those cases, though, there may be other options such as knockout models or indeed treated versus untreated wild type, etc., where the antigen is present in some, whether it's be treated or untreated, and not present in the other. Okay, I'll sum up just here. Hopefully this gave you some further insights. I feel quite strongly about running the appropriate positive and negative controls, that they're extremely relevant to get publication-worthy data. So please implement them. For those of you who may be looking for non-immune IgG, we do have some control antibodies at Vector Labs that you can use against the commonly used primary antibody species. So certainly look for those on our website. Please stay tuned for our next webinar, Maximizing Signal Retention in Immunofluorescent Applications, coming up in 3 weeks. So I'll finish up just now and I'll open the floor to any questions you may have. So thank you very much.
All right, thank you, Craig, for that informative presentation. We will now begin the question and answer session. Please feel free to submit your questions to the chats on this site. Okay, the first question is, what are the requirements for positive control tissue when the antigen has transient expression or is upregulated under certain conditions?
Yeah, so that sounds a little bit like a situation whereby you're unsure of the antigen being expressed there. How much is there, or possibly even if it is in that tissue? Maybe we can revisit that exception example that I showed, whereby look at the tissue type you've got there. Are there other targets you could look at to set up a positive control? I think the example I used in the presentation was if you're looking at an endothelial cell and maybe you've got transient expression, I think Byron mentioned there, or upregulated and you're not sure, look for epithelial expression. You could target a cytokeratin, and I think I also said then a smooth muscle or whatever it might be, and work up conditions for that. I understand it's not specifically what you're looking for, but the idea is to show that the tissue prep is appropriate, it's working, and the detection reagents. So if you get an absence of staining in that situation, at least therefore you're not double guessing whether or not the detection reagents are or are not working, if it's a sensitivity issue or whatever it might be. For that scenario as well, you're going to have to run a tighter series on that primary antibody against the target antigen. You're going to have to cut multiple sections. and you will put it on, let's just say a 1 to 100, 1 to 50, 1 to 25, whatever it might be, and see where that optimal signal to noise ratio does develop. But you want to try and optimize signal conditions. You don't want to not see anything. It's always better to see something, even background staining, and dial it down a little bit. But if you're still seeing nothing, that's the beauty of having something like a positive control, whereby you can leverage that to say, all of this is working, therefore either cut deeper into the block or whatever it might be to try and identify that known or that transient expression target antigen.
All right, thank you, Craig. The next question is, what is a better way to show specificity of an antibody, Western blot or a positive control?
I don't think that you can hold one above the other, in terms of validation of primary antibodies. And I know that there's certainly a lot of conversation about this out there in this field of tissue-based assays, immunohistochemistry and immunofluorescence. I think that they should be used in combination. Certainly, though, you've got to be careful in that some primary antibodies, depending on the conditions, denatured or non-denatured conditions in Western blot, they may not work for a native antigen in a tissue prep. So while it may work beautifully for Western blot, it might not work on a tissue that's been antigen retrieved with citrate, heated to 56 Celsius and embedded in paraffin or whatever it might be, and then exposed to other things such as potentially solvents like xylene and what have you. So, I think, hey, I would use as many data points as possible to validate that primary antibody, definitely.
All right, thank you. The next question is about pre-absorption. With pre-absorption, you are using an immunogen that was created for the antibody. Therefore, one is assuming that the immunogen is correct. Do you have any comments?
Yeah, certainly that would be certainly the scenario. In a highly defined, monoclonal antibody, for example, a purified preparation, I would say that is more than likely going to be appropriate. As I mentioned, though, the concern would be whether or not there may be, with say a polyclonal antibody, like residues or amino acid sequences on that tissue prep that the antibody may also prefer to bind to, or indeed whether there may be something that if you don't put that immunogen in at a certain concentration, there may still be availability of binding of that primary antibody to targets on the tissue site, or indeed while it works well in solution, maybe it's kicked off or eluted when you put it on the tissue prep and give some other staining there. So there's some of the caveats in applying that pre-absorption control. There are some considerations you have to weigh when putting it onto a tissue with a lot of different things on there, a lot of different proteins, macromolecules, charged particles that could potentially still attract that antibody or indeed cause elution, I said, of that target antigen. So just make sure that the affinity and avidity is good, and it does match some of the other data that we mentioned. This carries on, I guess, from the first question about validity of or validation of the primary antibody. Don't use it in a vacuum. Use it with other data that others may have generated about that antibody out there.
Okay, great. The next question is, for double immunolocalization, what are ideal positive or negative controls? Would this need to be done separately for each antibody?
In a quick response, one word, yes. So for positive controls, well, I'll expand on that a little bit. In that, yes. So I did hear it. It's co-localizing. It might be immunofluorescence, two antigens in the same prep. Make sure that the positive controls are in place. These will be run on separate tissue sections for that for each of those target antigens using the detection reagents you intend to use on that tissue prep. Now, the negative controls are a little different in that you'd have to run those separately, but also you will have to also run a negative control of, for example, running the first stain through to completion on the tissue prep, and then here's where you omit the primary for the second one, but put on the secondary detection reagents for the second one. Make sure that they're not binding to any part, could be a blocking reagent, of that first detection methodology. Brings me to another point in that if the two primary antibodies are mouse, if you're using two anti-mouse detection reagents, that's going to be problematic. So in that scenario, it'd be highly recommended to avoid that. Use a mouse and a rabbit primary antibody, or a rabbit and a goat, or a mouse and a goat, something like that, where that potential of cross-reactivity between the detection systems is eliminated.
Okay, great, Craig. Next question is about non-immune IgG. This customer is using the rabbit primary antibody, and their isotype control is producing high background. Their tissue is human placenta. Do you have any comments about that?
Yeah, so I'm going to revisit a point I made back in the first webinar, whereby it sounds like the isotype control you've got there is a little sticky toward that tissue you've got there. Certainly placental tissue, highly vascularized, a lot of protein floating around there, hormonal influences that could be causing some issues. What you can do is do an absorption in solution with that negative control with serum from that tissue type. So if you've got human placental tissue, you could use either human or non-human primate serum in buffer at about, use a 2 to 5% serum in buffer to dilute that control. Let it incubate for, say, 30 to 60 minutes. That should absorb out sticky components, and then you can put that on the tissue prep. If you do that with that control, you'll also have to do that with the primary antibody. So just ensure that the antibody, that the target antigen you're looking for is obviously not serum-based. But that it would certainly be an appropriate way to absorb up some sticky components in that scenario.
Okay, we have time for one last question. This customer is optimizing antibodies against human brain tissue. They're not seeing any staining now, and controls are run every time. Do you have any troubleshooting tips for no signal?
With that, I'm going to assume that the controls, particularly the positive control, is working. Which means that certainly the secondary detection reagents are valid, whether this be enzyme or fluorescent based. So it brings into question then either the primary antibody, is it cross-reacting with the target antigen of that species? That's one thing to consider. Another thing is the concentration of that primary antibody. Is it a lack of sensitivity? Certainly you could consider using a more sensitive detection methodology. You could hit it with a proverbial sledgehammer, hit it with a very sensitive detection system, more than what you've got there. If it's a secondary antibody conjugated to a fluorophore, there are other options out there. They take you a little longer, but at least that way you're going to see some staining that you can turn back on. So there are a couple of things you can consider. If the antibody is shown to work in Western blotting, I'll just go back to my point earlier about, what were those conditions for the Western blot? Maybe the primary antibody doesn't work for either paraffin embedded or frozen tissue sections. for that given species. So I would certainly have a look at that primary antibody, have a look at the sensitivity of the detection methodology you've got. That'd be key parameters that I would pinpoint.
All right, thank you, Craig. Unfortunately, our time is up for this webinar. We will send you the entire recording of this webinar soon via email. And if you did not submit your question or we did not follow up with you, we will answer them personally. And please feel free to send any additional questions to technical@vectorlabs.com. Our next webinar is on signal retention in immunofluorescence in 3 weeks on August 25th. We hope to see you there and have a great day. Bye-bye.
Thank you very much.